Hello everyone. In today's video, we are going to learn about gene therapy. So, why we are going to learn about gene therapy when we have already excellent conventional therapies available to treat the disease? We have uh, the varieties of modalities to treat diseases. We are treating it right now. So, why we are talking about this gene therapy, or what kind of treatment or what kind of disease we are going to treat with the help of gene therapy we will discuss today. So whenever individual suffering from genetic disorder that is inherited or the metabolic disorder that is inborn, once the patient is diagnosed with such type of disorders, then he or she lives on daily medication. He takes medicine to uh, like take care of the symptoms that is due to the disorders. That is a conventional treatment. There are certain drawbacks of this conventional treatment, like it gives a symptomatic relief only. It's a kind of palliative care. It's not going to cure the disease. It just delayed the symptoms and so on. So it has to be continuous for a lifelong. So that is what a conventional treatment help in a genetic disorders. To overcome this, what we can say a drawback of conventional treatment, we have to think about gene therapy. So gene therapy can provide treatment that is permanent cure and that gives a hope for a normal life that we not get with the conventional therapy when we talk about genetic disorders. This is the cover page of Time magazines, like almost 2 decades before, and you can see that the future of medicine, how genetic engineering will change us in the next century. And we are actually in that future. We are in the next century and we are getting the result of genetic engineering, and we are going to learn about the part of the genetic engineering that is gene therapy. So, what is gene therapy? The gene therapy is a technique for introducing a particular gene into existing cell to or prevent or cure a wide range of diseases. So, with the help of this, we introduce a gene. That gene undergoes a transcription that produces the messenger RNA, that undergoes a translation and produces the protein. And with the help of that, we can get rid of that particular diseases. There are different approaches for the gene therapies, like gene transplantation, that is for the patient having gene deletion, the patient who have not a gene, particular gene, that we can get the gene by the gene transplantation. Those who are having a specific gene mutation, we can revert it with the help of gene correction. Or the patient having a gene but it is not going to express because of somehow, then we can treat by the method that is gene augmentation. So let's talk about some history related to this gene therapy. The scientists have begun to think about the process of gene therapy almost before the 40 years, and like in 1980s. And the first gene therapy was performed after 10 years in 1990 for the patient suffering from severe combined immunodeficiency disorder that we all know because of adenosine deaminase deficiency. The patient was treated by transferring a normal gene for adenosine deaminase. That was the first case of in the history of gene therapy. Gene therapy, there are mainly two types. One is somatic gene therapy and the germ cell gene therapy. In somatic gene therapy, we are inserting the new gene into the somatic cells like fibroblast, myoblast, epithelial cells, nerve cells, and glial cells to treat that particular diseases. There are different two approaches. We will talk about that in detail. One is in vivo gene therapy, second is in vitro ex vivo gene therapy. The second type is germ cell gene therapy, in which we introduce a gene into a cell that is reproductive like in sperm or in the ovum, and it is considered as an unethical practice. So we are not going to talk about that germ cell gene therapy, and but we will discuss that somatic gene therapy. But remember, the somatic gene therapy is not goes to the hereditary. Okay, so the patient treated with the gene therapy, that will not go to the offspring. Okay, so we'll see how it works, the somatic gene therapy. For that, we first deliver the therapeutic gene that is defect or absent or not expressed in the patient's target cell. That gene will gain entry into the target cell with the help of viral vector. Viral vector will infect that cell with our therapeutic gene. The vector's genetic material will be inserted into the target cell DNA. So, in short, our therapeutic gene will get entry into the target cell DNA. This gene produces the functional protein. So, the therapeutic gene, as I told you, undergoes transcription that will go for the translation and produces our functional protein. This is the way how our cell return to a normal state. So this is the way how the somatic gene therapy works. So again, as I told you, like these are the viruses that act as a vector that carried our therapeutic gene and transfer it to the cell that is in nucleus, and that our therapeutic gene undergoes the process of translation and produces our therapeutic protein and make that cell a normal. So for this gene therapy, there are two approaches to deliver the therapeutic gene into the host cells, or what is a target cell. There are two different approaches are available. One is ex vivo gene therapy and second is in vivo gene therapy. In ex vivo gene therapy, the patient's cells, they are extracted from the patient and cultured in the laboratory. The new gene, that is therapeutic gene, will be infused into the host cells, and the modified cells, that is the patient's culture cell in which we have added our new gene, that modified cell will be administered back to the patient. That is ex vivo gene therapy. The second is in vivo gene therapy. We'll see it later. This is ex vivo gene therapy. As I told you, the patient's cells are harvested. We take virus that is altered in the laboratory so it cannot reproduce. So there is no chances of getting infection of that particular virus. Later on, we will insert our therapeutic gene to this virus. Our harvested cell with the virus that is contained therapeutic gene, that will be mixed, they will be grown together. So this viral genome will be integrated into the human genome, and then it will be again injected, into the patient's body. That cell with a viral genome will undergo the translation and it produces the desired protein. So this process in which we are taking the cell from the patient, we go through the procedure that is outside the body, and then we inject that altered cell into the patient's body. This is why its technique is called ex vivo gene therapy. The second approach is in vivo gene therapy, in which the genetic material, or we can say a therapeutic gene, is directly transferred into the body of patient, where the vector is administered directly to the cell, an only available option for the tissue that cannot be grown in vitro. Okay, so that is the available option only for those tissues that cannot be grown outside the body. In vivo gene therapy, there are varieties of vectors like a liposome, a recombinant virus, or the DNA, or plasmid or cosmid that will be directly injected into the patient by the IV fluid, or in the form of injection, or with the help of ballistic gene gun, or like this CFTR gene defect in cystic fibrosis, that therapeutic gene is transferred directly to the lung. So these are the techniques, or the methods in which we directly introduce that gene into the host cells, and that is the in vivo gene therapy. So to transfer the therapeutic gene, or what can say target gene, to the host cell, we require a carrier, that is vector. So this is the way to insert normal gene in the patient's cell, is through vector. The most common vectors that are used in the gene therapy is viral. There are list of viruses that we are using treating the patient with in gene therapy, but there are certain non-viral vectors are also available. The viral vectors, they are called the biological vectors, and apart from that we have physiological vectors as well as chemical vectors, that are the liposomes, the naked gun, there's a naked DNA, and we can do it with the help of, as I told you, uh, ballistic gene gun method. So why we are using viruses as a vector in the patient treating with the gene therapy? Because through the time of evaluation, through the time of evolution, we know that viruses can be able to infect the cells with a very great specificity. They tend to be very efficient at transferring their own DNA into the host genome. So their viral gene easily get incorporated into the human cell genome, and this allow them, by this mean incorporating that viral genome into the human genome, they allow them to produce new viral particle at the period of cell synthesis. There is a list of viruses that are used as a vector in gene therapy. There are retroviruses, adenoviruses, lentovirus, pox virus, herpes virus, and so on. They have their different properties. Based on that, they are used. They have different properties to infect a particular cell in the human being. Based on that, they are used in the gene therapy. Let's talk about the retroviruses. The retroviruses are RNA viruses. We know that, like an HIV viruses. So they create double-stranded DNA copies from their RNA material by the reverse transcription process using reverse transcriptase enzyme. Then by the reverse transcriptase, this RNA genome convert it into double-stranded gene, and that will get entry into the human genome using integrase enzyme. Now the issue is that the integrase enzyme introduced that double-stranded DNA into the human genome, but it cannot insert that gene at the specific site. So it may be inserted at somewhere else, and that may produces insertional mutagenesis. So instead of we are getting the therapeutic effect, we get the effect of mutation. So vector used that derived from the human immunodeficiency virus and are being evaluated for safety. This is the most common virus. HIV virus is used it's what we can say gene therapy as a vector. Advantage of retroviruses: the integration, they produces permanent expression. They're pseudotype viruses. So once we introduce these viruses, they're no more infective. They just transfer the genetic material or DNA into the host DNA. The disadvantage is the retrovirus only act on the dividing cells. So that is the limiting factor using the retroviruses. While we can use adenoviruses, those the tissue or the cells that are non-dividing. This is one of the important thing that we have to think when we use retroviruses, that is insertional mutagenesis. So this gene which we want to introduce into the human DNA, it may insert at such place that it may activate oncogene or inactivate or inhibit the tumor suppressor gene and may leads to cancer formation. That is the one of we can say a limitation of this retroviruses or the virus as a vector. So here you can see a virus that contain a DNA, that is modified DNA. Here you can see in a viral DNA in between a therapeutic gene is inserted. Then we infect this cell with that virus, that will get entry inside the cell, that will get attached to the nucleus, and this vector inject the gene, that is therapeutic gene, into the nucleus. This gene will get entry inside the nucleus, then it incorporated with the host DNA that will undergoes transcription, translation, and produces the therapeutic protein. So this is the way how viral vectors act. The advantage: they offer high titers, and easier ability to infect wide varieties of cell. But the disadvantage: the expression is usually uh, transient and the useful effect varying from a few months to a few weeks to a few months. That is a disadvantage of these viral vectors. Now, as I told you in the beginning, there are viral vectors as well as there are non-viral vectors. The non-viral vectors have potential to avoid some critical problems or side effect what we are getting when we use viral vectors, like immune responses. After all, they are a kind of viruses. So our body's immune response may try to fight with that vectors. The viral vectors have a limited packaging capacity, and as I told you, they may able to produce insertional mutagenesis because of their random integration. To avoid this, we may use non-viral vectors. The first one of that is a liposomes. The liposome mediated CFTR gene transfer was used in the patient for the cystic fibrosis. The liposomes, we know they're nothing else but artificial lipid bilayer. In the cavity of that lipid bilayer, we can incorporate a plasmid carrying our therapeutic gene. The complex that get entry inside the target cell by fusing the cell membrane or the plasma membrane. The liposome may be mildly effective, but their activity does not last long. And there are cationic liposomes that means positively charged liposomes, so they can get easily attached with the DNA that are negatively charged. There are certain advantages of liposome using as a vector that can carry a big size of human gene that cannot be done with the viral vectors, and it evoke a very weak immune response. So there is a less chances of rejection. But the thing is that the disadvantage of this liposome using as a vector is most of the complexes when we introduce, they are destroyed by the host cells, and that reduces the efficiency of this therapy. This is the physical method using or the introducing the therapeutic gene into the target cell with the help of biolistic DNA injection, that is also known as gene gun. It is invented for DNA transfer to the plant cell, but it is fully applicable to the eukaryotic cell also. But third, we have seen the process of gene therapy. It has few problems that we have to overcome over a period of time. Like, it has short-lived nature of gene therapy. It is very hard to achieve long-term benefits without its proper integration. So it is one of the drawbacks of gene therapy. If it is not integrated properly or to the particular cell, we will not get the advantage of that gene therapy for a long time. Immune response. It reduces the gene therapy effectiveness and makes repetitive rounds of the gene therapy and make it useless. The problem with the viral vectors, there are chances of toxicity, immune and inflammatory responses against that virus, or there may be a possibility that viral vector may reactivate and may cause the diseases. The thing related to gene therapy is that we can treat the disorders that is due to single gene defect. There are nowadays what we can say trial going on to treat the disease having multi-gene defect, but the most commonly occurring disorders, they're like heart disease, Alzheimer's disease, arthritis, diabetes. They are the multi-gene disorders. They usually cannot be treated or not treated easily with the help of gene therapy. So though there are problems, there are some issues with the gene therapy, but we have a long story of success to tell about the gene therapy. There is a list of disorders that can be treated with the help of gene therapy, like SCID, severe combined immunodeficiency disorder. That was the first disorder treated with the gene therapy. Then DMD, that is Duncan muscular dystrophy, cystic fibrosis, hemophilia, cancer. Not all the cancers, but there is a certain list of cancers that can be treated with the help of gene therapy. Familial hypercholesterolemia, Leber's hereditary optic neuropathy, Parkinson's disease, sickle cell anemia, and so on. A very long list we can get with the help of the list of disorders that we can treat it with the help of gene therapy. So this was the list of disorders that we can treat with the help of gene therapy. This is the photographs of a patient that was first time treated with the gene therapy, that is Ashanti de Silva in 1990s. She was diagnosed with the SCID due to adenosine deaminase deficiency, and with the help of gene therapy, the therapeutic gene, that is adenosine deaminase enzyme, which introduced into the patient, and then she survived very well. So the conclusion at the end of the session, that there are ethical issues involved with the gene therapy, but with the time we have to resolve it. The gene therapy is still new and experimental, though it's almost more than 3 decades in the gene therapy, but still we can say it's a new and experimental. We have still a very long way to go to treat various diseases, that is a single gene defect as well as to the multi-gene defect. Theoretically, we can say it's a permanent solution of any genetic disorders, but the thing is, it has to give a lifelong expression of that particular gene. In future, the day may come when almost every disease will have a gene therapy. So this is the hope we are waiting for. Thank you so much. So this is all about gene therapy. For more updates, please follow us on Insta page, that is at the red biochemistry for student, and like, share and subscribe for more videos. Thank you so much.